Now here is a competency based question. Why is the rate of breathing in aquatic organisms much faster than in terrestrial organisms? It means why there is a difference in the rate of breathing between aquatic organisms and terrestrial organisms. Students, remember the rate of breathing in aquatic organisms is faster because the amount of dissolved oxygen in the water is lower as compared to the amount of oxygen in air. Aquatic organisms like fishes, they obtain oxygen from water which is present in dissolved state through their gills. Since the amount of dissolved oxygen is fairly low compared to the amount of oxygen in the air, hence the rate of breathing in aquatic organisms is much faster than the rate of breathing in terrestrial organisms. Dear students, here is a variegated plant. Example, money plant. Actually, these variegated plants have leaves that are partly green and partly white. By testing these leaves for the presence of starch, we can determine which parts of the leaf have undergone photosynthesis. You know, green areas contain chlorophyll, so only green areas will absorb the sunlight and perform photosynthesis and manufacture glucose, which ultimately gets stored in the form of starch. So dear students, for all the three experiments of photosynthesis, the first step is take a potted plant and place it in the dark for 3 days, that is equal to 72 hours. So placing the plant in the dark ensures that any starch present in the leaves is used up. Next, after destarching, we will place the plant in sunlight for few hours, maybe 4 hours or 6 hours, and allow the plant to undergo photosynthesis. Next, what do? We pluck one leaf and then boil it in water for few minutes so as to kill the leaf. We will boil the leaf in alcohol using a water bath until it becomes decolorized. So the purpose of boiling leaf in water bath is to remove all the chlorophyll. Otherwise, when we do iodine test, the blue-black color will not be clear. The chlorophyll will interfere with the iodine test. That is the reason we need to boil the leaf in alcohol using a water bath. And one more important point here is we need to boil the leaf in alcohol using a water bath, but not directly, because alcohol can catch fire and can cause accident. So to avoid that, we place a small beaker having alcohol into a big beaker having water. Then we boil it so that accident does not occur. Next, after boiling in alcohol using water bath, we will wash the leaf in water and then do the iodine test. So when we do iodine test, the green parts of the leaf which contain chlorophyll will turn blue-black, indicating the presence of starch. Whereas the white parts of the leaf which lack chlorophyll will not change color, indicating the absence of starch. This demonstrates that chlorophyll is essential for photosynthesis. Now students, the next image is about the experiment to show that carbon dioxide gas is essential for photosynthesis. Most important for board exam. So students, in this experiment, to show that carbon dioxide is essential for photosynthesis, we take two healthy potted plants almost of the same size. Next, place the plants on separate glass plates. Then cover both the plants with separate bell jars. We keep a petri dish containing potassium hydroxide and place it inside one of the jars. So what is the purpose of the chemical potassium hydroxide? So as to absorb the carbon dioxide gas that is present inside the bell jar. And here the most important precaution in this experiment is the setup must be airtight. So we need to seal the jars with Vaseline. Then we place the plants in sunlight for 2 hours. Then after that, we remove a leaf from each plant and check for the presence of starch as done in the previous experiment. Pluck a leaf from each plant and test for the presence of starch. Before we perform iodine test, first both the leaves have to be boiled in water, then in the alcohol using water bath. Next, wash the leaves in water, then do the test with iodine. What do we observe? See, in setup A, KOH is placed which has taken up all the CO2 gas. Due to non-availability of CO2 gas, plant A cannot perform photosynthesis. Hence, it does not show blue-black color when tested with iodine solution. The leaf of the plant in setup B kept without potassium hydroxide will turn blue-black. Due to the availability of CO2 gas, the plant will perform photosynthesis, hence gives positive test for starch. Whereas the leaf of plant in setup A does not show positive test for starch test, indicating that CO2 gas is necessary for photosynthesis. Next question, name the structures which are present on the bark of the tree which help in exchange of gases. Now here these structures are named lenticels. They are found on the bark of the trees. Essentially, lenticels are specialized pores that facilitate gas exchange between the plant's internal tissues and the atmosphere. Next experiment to show that sunlight is essential for photosynthesis. Here also we take a potted plant with green leaves, place it in dark for 3 days so that all the starch is used up. Next, cover the part of the leaf with black paper. Now keep the plant in sunlight for about 6 hours. Then pluck the partly covered leaf from the plant and remove the black paper. Next, boil the leaf in water, then in alcohol using a water bath. Next, wash the leaf with water, then test with iodine solution. What do you observe? You will observe that the uncovered portion of the leaf has absorbed the sunlight and performed the photosynthesis. Hence, it will turn blue-black when tested with iodine solution. The covered portion of the leaf will not show blue-black color when tested with iodine solution because it cannot absorb the sunlight, hence cannot perform photosynthesis. This proves that sunlight is essential for photosynthesis.